Welcome back, everyone. Ready to take another deep dive with us? Today, we're going to be tackling mass spectrometry, and I'm really excited about this one. We've got a whole stack of articles and research ready to go. And of course, we've got an expert here to help us sort through it all.
I'm happy to be here.
So for anyone just joining us, let's set the stage a bit. Mass spectrometry, it's all about measuring the mass-to-charge ratio of ions, which gives us those unique molecular fingerprints.
Yeah, exactly. And what's really amazing is that it doesn't just identify molecules, it quantifies them too. So we know what's there and how much of it.
That's so cool. And to do all that detective work, we have these amazing instruments called mass analyzers. The articles we've got here focus on four main types: quadrupole, time of flight, magnetic sector, and ion trap. And I have to say, each one is like its own little world of science.
You could say that each analyzer has its own strengths and limitations, kind of like specialists on a team. Choosing the right one really depends on the problem you're trying to solve.
I love that analogy. So let's start with the quadrupole analyzer, often called the workhorse of mass spectrometry. Why is that? What makes it so popular?
Well, it's really reliable and versatile, for one thing. They're also relatively simple to use and maintain, which is a huge plus in the lab. Think about things like drug testing for athletes or analyzing pesticides in food. That's where the quadrupole really shines.
Wow, so it's really behind the scenes in so many fields I was reading that it basically creates this electric field obstacle course for ions, right?
Yeah, that's a good way to visualize it. You have these four parallel rods that create an oscillating electric field. Only ions with a specific mass-to-charge ratio can navigate that field and make it to the detector.
So it's like a molecular sieve, letting only certain ions pass through. There was also something about a stability diagram. What exactly is that?
Ah, yes. The stability diagram is basically a map that shows which ions will have stable trajectories within the quadrupole. By adjusting the voltages on the rods, scientists can target specific ions. It's kind of like tuning a radio to a specific frequency.
Okay, that makes sense. So scientists can create a custom filter for the molecules they're looking for. But I imagine even a filter has its limits.
You're absolutely right. One limitation of the quadrupole is its resolution. It can struggle to distinguish between ions with very similar mass-to-charge ratios.
So what happens when you need to analyze complex mixtures or look for really tiny differences between molecules?
Well, in those cases, you might need a different type of analyzer, something like the time of flight or TOF analyzer. This one takes a very different approach.
And from what I've read, speed's the name of the game with TOF, right?
Exactly. Think of it like a race. Ions are accelerated through an electric field, and the lighter ions, being more nimble, will reach the detector first. By measuring the time it takes each ion to travel that distance, we can figure out its mass-to-charge ratio.
Okay, that makes sense why it's called time of flight. But wouldn't the initial energy of the ions mess things up? Like, wouldn't some get a head start?
That's a great point. And that's where the reflectron TOF analyzer comes in. It's like a mirror at the end of the racetrack, reflecting ions back and correcting for those energy variations.
That's pretty ingenious. So the reflectron makes sure all ions with the same mass reach the detector roughly the same time, regardless of their initial energy.
Precisely. And because of that, reflectron TOF instruments have much higher resolution than standard TOF analyzers.
So it's like a high-definition version of the TO analyzer. I also came across something called orthogonal TOF. Is that just a fancy name for the same thing?
It's a variation, yeah, but with a bit of a twist. In orthogonal TOF, the ions are sent into the flight tube at a right angle to the accelerating field.
So it's like adding another dimension to our molecular race. What's the advantage of doing that?
It lets us separate ions not just by their mass-to-charge ratio, but also by their velocity. So it's like getting two pieces of information for the price of one.
That's impressive.
Mhm.
I can already see how valuable that would be in research. Before we get too far ahead, though, let's talk about those applications. Where does the orthogonal TOF really shine?
One of the most exciting applications is in imaging mass spectrometry. It allows researchers to visualize the spatial distribution of molecules within a sample. It's like taking a snapshot of the molecular landscape.
That's incredible, like mapping the different components of a tissue sample or even a single cell.
Exactly. And this leads us to an important point: the importance of high resolution, especially in TOF analysis. Why do you think it's so important?
Well, I imagine if you're trying to distinguish between molecules with very subtle differences, you need that level of precision. Like if you're looking at different isotopes of an element or trying to spot tiny modifications in a protein.
That's exactly right. Without high resolution, those subtle differences could be completely missed. Now, let's move on to the third type of mass analyzer on our list: the magnetic sector. It's often described as the classic powerhouse of mass spectrometry. What do you think earned it that title? It's got a long history, that's for sure. Magnetic sector instruments were some of the first mass spectrometers ever developed, and they're still known for their accuracy and resolution. Really impressive stuff.
So they're like the pioneers of the mass spec world,
Yeah,
still going strong after all these years.
you could say that, but don't let their age fool you. They're still incredibly powerful.
Okay, so how do they actually work? What's the secret sauce that gives them that high performance?
Well, it all comes down to using a magnetic field to separate ions. It's based on their momentum. Picture ions traveling along a curved path. Heavier ions have more momentum, so they follow a gentler curve, while the lighter ions take a tighter turn.
It's like a molecular racetrack. The heavier cars stay on the outer lane while the lighter ones zip around the inside.
That's a good analogy. And by measuring the radius of that curve, we can figure out the ion's mass-to-charge ratio very precisely, too.
But wait, wouldn't the ion's initial energy also affect how it moves through the magnetic field?
You're right, it would. And that's where the double focusing analyzer comes in. It combines a magnetic sector with an electrostatic sector, which acts like an energy filter. So all the ions entering the magnetic field have the same kinetic energy.
It's like making sure all the cars on our racetrack start with the same speed.
Exactly. And this double focusing design is what gives these instruments such incredible resolution and accuracy.
So that's the key. Now, where do we really need that level of precision? What kind of research relies on it?
One area that comes to mind is geochronology. It relies on super precise measurements of isotopic ratios to figure out the age of rocks, fossils, even ancient artifacts.
Wow, so they're helping us piece together the history of the planet.
Absolutely. Everything from the formation of the Earth's crust to the evolution of life itself, magnetic sector instruments are playing a big role.
That's pretty amazing. It's fascinating how mass spec can answer these fundamental questions about our world.
It really is. But even these powerful instruments have their limitations. Their complexity makes them a bit more challenging to operate and maintain compared to, say, a quadrupole.
So there's a trade-off between power and practicality.
Yeah, you could say that. Another thing is compatibility with certain ionization methods. Double focusing analyzers aren't as well suited for pulse techniques like maladia, which are common in biological research.
So like you said earlier, it's all about choosing the right tool for the job.
Exactly. No one size fits all in mass spectrometry. Now, let's move on to our final mass analyzer, the ion trap. This one is really unique in how it approaches ion analysis.
I'm all ears. From what I read, ion traps actually capture and hold ions for a while, almost like tiny molecular prisons.
That's a great analogy. It's a big difference from the other analyzers we've talked about, where the ions just fly right through. Ion traps use oscillating electric fields to confine ions in three-dimensional space.
So it's like a holding cell where you can really study those ions in detail.
Exactly. And just like the quadrupole, we have a stability diagram for ion traps. It shows us which ions will stay trapped based on their mass-to-charge ratio and the applied voltages.
So scientists can really fine-tune the trap to catch specific ions.
Right. And once you've got those ions trapped, you can do all sorts of things. You can eject them based on their mass-to-charge ratio for analysis, or you can even do multiple stages of mass spectrometry, or MSMS, right there in the trap.
Hold on, multiple stages. What does that even mean?
Think of it like an interrogation room. In MSMS, you first isolate the ion you're interested in. Then you break it into smaller pieces. By analyzing the masses of those fragments, you can learn even more about the structure of the original molecule.
Just like cracking a code.
Exactly. And this ability to do MSMS is a big advantage of ion traps. They're super versatile, especially when you need to figure out the structure of complex molecules.
I bet that's invaluable in fields like proteomics, where researchers are trying to identify and sequence peptides.
Absolutely. Ion traps are essential tools for that kind of work, and their versatility goes beyond MSMS. They're also used for gas phase ion chemistry experiments, studying how ions react with other molecules in a controlled environment.
So it's not just about identification and quantification. It's about understanding how those molecules behave and interact.
Precisely. Ion traps give us a glimpse into that dynamic molecular world.
That's pretty amazing. But I guess like any technology, there must be some downsides to ion traps too. What are some of their limitations?
Well, one challenge is that they're prone to space charge effects. That happens when too many ions build up in the trap. It can distort the electric fields and mess with the accuracy of the mass measurements.
It's like having too many people in a small room. Things get chaotic.
Exactly. And that crowding can affect the sensitivity and resolution. Another limitation is their mass range. Ion traps generally have a lower mass range compared to some of the other analyzers.
So they're not ideal for studying really big molecules.
Not really, no.
Yeah.
But for analyzing smaller molecules and peptides, they're incredibly powerful tools.
That makes sense. Well, we've covered all four types of mass analyzers now. It's incredible how each one has its own strengths and weaknesses. As we wrap up, what are some of the key takeaways you want our listener to walk away with? What's the big picture here?
Well, first and foremost, I hope they appreciate just how powerful and versatile mass spectrometry is. It's constantly evolving and finding new applications. It seems like every field is using it in some way.
It's like a Swiss Army knife for molecular
I
analysis.
like that. And second, I want them to understand that there's no single best mass analyzer. Each one has its own pros and cons, so choosing the right one really depends on the research question you're asking.
It's about assembling the right team for the mission.
Exactly. And finally, I want our listener to remember that mass spectrometry isn't just about identifying and quantifying molecules. It's also about figuring out their structures, understanding how they interact, and really digging deep into that molecular world.
It's like we're opening up this invisible universe of molecules.
That's a great way to put it. And that's what makes this field so exciting. There's always something new to discover.
Okay, I have to ask, what are you most excited about in terms of the future of mass spectrometry? What's on the horizon that really gets you fired up?
Oh, there's so much to be excited about. But one area that really stands out is the development of miniaturized mass spectrometers.
Miniaturized. Like how small are we talking?
Think portable.
Yeah.
Maybe not quite smartphone sized yet, but we're getting there. Imagine handheld mass specs that could be used for on-site analysis in all sorts of fields.
That would be game-changing. What kind of applications are we talking about? Give me some examples.
Environmental monitoring, for one. You could instantly detect pollutants in the air or water, or food safety. Imagine testing for contaminants or verifying the authenticity of products right there in the grocery store.
Wow, that's incredible. Real-time molecular analysis at our fingertips.
Exactly. And the potential for healthcare is huge. Think about handheld devices that could diagnose diseases with a simple breath test or monitor patient responses to treatment in real time.
It would revolutionize healthcare, that's for sure.
Another area that's really exciting is the integration of mass spectrometry with other techniques like microscopy.
So combining different tools to get an even more complete picture of the molecular world.
Exactly. By coupling mass spec with microscopy, we can not only identify the molecules in a sample, but we can also visualize where they are. Spatially, I mean.
Like adding a GPS to our molecular detective kit.
That's a great way to put it. And we're already seeing some amazing discoveries from this combined approach. Researchers are using it to study the inner workings of cells and tissues, mapping out different proteins and lipids. The level of detail is incredible.
It's mind-blowing how much we're still learning about the basic building blocks of life.
And it's not just biology. This integrated approach is being used in materials, science, forensics, even art conservation. It's really everywhere.
A truly multidisciplinary tool, it sounds like. But for our listener who might be thinking, okay, this is all cool, but how does mass spec actually affect me? How would you answer that?
Well, mass spectrometry is working behind the scenes in so many ways that impact our daily lives. Take environmental monitoring. It's used to detect pollutants in the air we breathe and the water we drink.
And it's playing a crucial role in food safety, making sure the products we consume are safe and authentic.
And in healthcare, it's helping diagnose diseases, develop new drugs, even personalized treatments based on an individual's genetic makeup.
It's even used in forensics to solve crimes. So even if we don't see it, mass spectrometry is constantly making our world safer, healthier, and more sustainable.
It's an invisible force for good. You could say that.
Well said. As we wrap up this deep dive, I want to leave our listeners with one final thought. Imagine a world where we could instantly analyze the molecular composition of anything around us. The food we eat, the air we breathe.
Imagine detecting early signs of disease with a simple breath test, or pinpointing the origin of a mystery substance with a handheld device.
It's a future where the invisible world becomes visible, empowering us to make better decisions about our health, our environment, our world.
And that's a future that mass spectrometry is helping to build.
And that brings us to the end of our deep dive into the world of mass spectrometry. We hope you've enjoyed the journey and learned something new along the way. Until next time, keep exploring, keep asking questions, and keep diving deep into the wonders of science.